right.
Okay, thank you very much for coming. It's pretty loud. I think it's okay, the mic. Let me try to tell you what I know about computational biology of aging, mostly from our own experience. So for that, I will tell you a little bit how much, well, maybe the overall title will be like, we're living in the best of the times, meaning that suddenly we have lots of data on aging animals. 10 years ago, we had most of the data of aging animals, about aging animals from mice studies, but I think 3 to 5 years to now, we are in the situation for the first time in history where humans became the most studied animals in the world. We have hundreds of thousands of electronic medical records in a single country, like in the US, a lot more in other countries. We have tens of millions of people genotyped. The genotyping technology improves, which means that suddenly we are able to study aging and diseases in humans first. So basically, I will tell you that nature is running clinical trials for us. We are mutants, we are working mutants. We have thousands of genes knocked out in each of us, meaning that some of those genes are helping some of us, not helping others. Some people have less diseases because of that, and we hope that some people have less aging because of that. And what we need to do now is to combine all these magic powers that we're getting to understand what are those possible molecular targets for future interventions that would help us to cure aging and disease. So I will show how these things actually work in mice and humans from actual calculations. I will tell you how many agings are there in different animals and which of those are more important for longer lifespans. I think that will be kind of a demonstration how much, well, in machine learning we are learning, but in fact we want to unlearn aging. So I will tell you how to unlearn aging from big biomedical data. And finally, I will try to combine all of that and reflect on the difference and relation between aging and diseases, because as you know, you earn money, you have to cure disease. I will try to tell you maybe how to combine aging studies and studies on disease, and that would hopefully be beyond the time I have to speak. So how to fund the dream will be the last part. So if this works, I will, well, normally I talk quickly, so let me know if I'm talking too quickly. So just to give you the idea how we ended up here. So 1,000 years ago, I was presented to this guy. Actually, I've seen these guys, in the lab. So these wonderful creatures, naked mole rats, I was told, are not aging. Actually, I graduated from physics department. I had a decent job. I was doing theoretical condensed matter physics, and then somebody told me that there are guys that are not aging, and life has never been the same. So what happens is that normally, if you're a human, your chances to die of all causes go up exponentially and double every 8 years. Think about that. Even your chances to get killed by a bus actually go up and double exponentially every 8 years, because as we know, a pedestrian path, well, city environment is not dangerous for a little guy, but it's getting dangerous for elder individuals because of neurodegeneration problems. So in these animals, and this is the actual figure from the work by Google. Just think about that. In order to study non-aging, you have to build Google first, earn money, and spend this money in order to prove that these animals do not age. So that was insignificant discovery for the aging community. So what happened here is that the guys plotted the chances to die as a function of age for these animals for 30-something years. And you can see that, of course, there is some error margin because fewer and fewer animals are alive, but there is no exponent. This is not even a logarithmic plot. So the chances to die for these animals are the same all the time for 40 years. In humans, you have 5 doublings. So the chances to die for humans go up 32 times over this time scale. So these guys don't know how old they are. And this is, in my view, a criminal negligence because it turns out that in nature there are many animals that do not age. More than 1,000 animals are suspected to be not aging. You don't know if they're aging or not because you have to wait, thousands of years to find out. But the interesting thing is that there are animals that do not age, and they are mammals. and I'm also mammal, last time I checked, so this is an example. So the other thing why I am here is that there is a guy, Stuart Kaufman. He was trained as a doctor, but he became a theoretical physicist. This is an exceptional example. And this guy was studying, what I will tell you what is that, the stability of different genetic regulatory networks. And when the first transcriptomes, the first omics, right, because I'm telling you about computational biology, so I have to use the word omics. So when the first transcriptomes, the first signals from cells of different aging animals showed up, he tried to study how regulatory networks, how the ways the interactions between different components of the networks are organized. From the engineering science perspective, he was trying to understand if those networks amplify the noise or not, if they are stable or not. And the conclusion of his paper was that for each of the examples, from the experiment, with experimental accuracy, he could not find out if those guys are stable or not. So all living species, all living creatures, turned out to be operating just at the boundary between stability and instability. So when I've seen that, and when I've seen that, I decided to think that maybe those who are unstable are aging, and those who are stable are not aging, and then the goal of the computational biology in this example, computational biology is huge, but I will give you just one example, is to actually to understand which animals are just exponentially disintegrating, exploding and die, and which animals are just stable and don't want to die. So basically that was the plan. The idea was to study how the stability properties of different animals are different in different species. So what is stability? So as I told you, the stability is the property of amplifying the noise. Imagine that this is, I think, an example that is intuitive for everyone, a nuclear power station or a nuclear bomb. So in a nuclear power station, you have all those errors like neutrons, particles that are hitting the nucleus. The nucleus split, produce more neutrons, and since there are lots of materials that absorb the neutrons, there is no chain reaction. So this thing produces neutrons, produces heat, and it's stable and produces heat. Nobody is killed. Lots of power and steady state with some fluctuations of neutrons. In a nuclear explosion or in a bad nuclear power plant, you have a situation where some of those materials are not absorbing neutrons anymore. And then neutrons producing neutrons, and even more neutrons are producing even more neutrons, and you get exponential amplification of neutrons, and you have the explosion. So basically the idea was to use the actual biological data and to find out if errors, regulatory errors, mutations, whatever damage is there, is staying under control in some animals, and then you have just fluctuations of the organism state, whatever you measure there. So these guys are stable and not aging. And if there are animals where the damage is not controlled, I mean, these guys evolutionarily decided not to waste their time for controlling damage. And then these animals are exponentially disintegrating, and you have aging guys, you have non-aging guys, and the most important question was, where are the humans on this timescale, on this picture? So we set up a company to actually do that. I mean, the company is really organized around this idea. And we got the data, we started publishing, get a lot more data, published more, got a deal with Pfizer. Pfizer gave us a lot more data, which means that now we have access to millions, tens of millions of electronic medical records, including biomarkers for humans, aging humans, humans with disease and everything. And I will tell you how to use all that information, along with the genetic, molecular data, everything, to actually learn how aging occurs in humans, rather than try, to get the best examples from mice or somebody else. So what we can learn. So once again, aging is not very simple. If it were simple, we would not be here, because it would be already solved. So I have to show some pictures that, well, at least colorful. So basically, we are looking at universal features, and I will be switching between different data sources. I would be talking about blood markers, I would be talking about genetics, I would be talking about electronic medical records. Just for you to understand is that when we're talking about aging, we are looking about massive changes that occurs everywhere, on every level of the organization, which means that whatever I am telling you actually occurs in the same way on different levels. So you see the same graphs, no matter what you are watching. So you have lots of measurements. This is just an example. So these are measurements, blood markers from mice. That's the same things that you are getting when you are going to a doctor red blood cells, white blood cells, I mean, easy stuff. It doesn't need to be genomics, doesn't need to be transcriptomics. We all know that certain features go together. So, for example, if you plot the correlations between features of the white blood cells, they are correlated. If you are talking about red blood cells, they are correlated with hemoglobin levels and everything else. So always in biological signals, you have lots of correlations, which means that there are factors, there are much fewer factors than you have measurements that are going together. One factor is controlling your oxygen supply, the other factor is controlling your immune system, and so on. So you have clusters of features. And it doesn't matter, you can have the whole transcriptome and there would be clusters of features. Interestingly, that almost all the time, if you compute the mean features in this cluster, the mean activation of features in this cluster and these clusters, and if you plot them as a function of age, you would find that only one cluster of features normally depend on age. So aging most of the time is most of the time the largest cluster of correlated features, but this is only one cluster of correlated features. This is the example from mice. Actually, the variance is increasing. There is one cluster of features that is going up, and this must bring you a simple intuition, by the way. So I don't know if you were reading about things like catastrophes, like energy blackouts in energy grids, or catastrophes, or the meltdowns in financial markets. So people have been thinking forever, what are the signs of future disintegration? And the signs of future disintegration is that right before the catastrophe, lots of features become correlated and the whole system becomes slowing down. So there are two red flags. One of them is called critical fluctuations, where the system becomes uber correlated, which means very few variables actually need to describe it. And the other is that it's becoming slow. When you see these two features, most probably the system in question decides to disintegrate. So what we're trying to do here is that we are taking the biomedical data from animals that is organized in time series. So we're looking at trajectories. So we're looking at measurements that are taken at equal periods of times. And since I told you that there is only one feature that is associated with aging, we train artificial neural networks that combine all the features, doesn't matter how many features we have as inputs, into a single variable that is used to predict the same single variable in the future. So we're building very simple future telling machines that are taking the health state of an animal, of a human right now, compress it into a single variable, and then they use that single variable to predict this variable in the future and decompress this variable into the medical observations. So if you can do that, then this variable, the most predictive variable that is predicting your future health state from the current state, would be what? That variable would be the natural biological age. So if you do this, if you run this algorithm for a mouse, and this is the actual calculation, you measure this inferred variable as a function of age in mice, and you find that this variable is going up exponentially with age, exponentially. The exponent is exactly the same as the exponent in mortality acceleration, so this is the exponent. And the interesting thing is that the red dots here on this graph are the mice, because these are laboratory measurements. So these mice are scheduled to be killed in the laboratory because they are too sick. So you see that artificial intelligence actually works like ethical committee. So in laboratories, mice actually die due to limiting biological age. So you can see it doesn't matter what is the age of these animals. When they hit the ceiling in biological age, there is a good guy or a girl from the ethical committee coming and killing these guys. In aging field, by the way, the problem is not you have to fight with the ethical committee to let you keep those aged animals alive to study aging, because this is presumed to be unethical. So the takeaway from here is that you can use these are called autoregressive models. For those who care about large language models, they are also autoregressive models. They are unsupervised. You simply require that from the current state you predict the future state. It's like you predict the future health state of the organism. And from those, if you do it by looking at the physics textbook, you find variables that are immediately interpretable. So that's how aging looks in mice. Now, The interesting thing is that there is no time here. You have lots of features that depend on age, but in fact, they only depend on the biological age. There is no atomic clock in mice or in you. If there were such an atomic clock, then of course biologists or doctors would take that atomic clock out and we would be living forever. So actually the only time variable that exists in these animals is this biological age. And here, for example, we plotted the number of senescent cells in mice versus our biological age, which was inferred by the network. And you can see that the variables that were not fed into the network still correlate to the biological age. So this is called the correlation between different hallmarks of aging. So senescence is a hallmark of aging, loss of your red blood cells is your hallmark of aging. So all of them are correlated, remember I told you. So if they are correlated, it means that you can use just one variable instead of many variables, and that variable is the measure of aging. So that's how you get the biological age. That also means that if you reduce this biological age by a drug, all hallmarks of aging would also get reduced. And I will show you in a moment that it actually works like this. So I always like to talk about computational things immediately in conjunction with the experimental data that we're having. So instability, so mice are unstable, they are, as you can see, exponentially disintegrate. Instability may look scary, but if you are unstable, it means that there is no stable state that you can return to. Which means that if you have lots of damage and then somebody is coming and reduces the damage, you should keep aging, but from a lower base level, if this theory is correct. So essentially, I believe that if you have an aging animal and your biological age is going up, if you do a treatment that reduces the biological age, you should produce rejuvenation. So animals would never catch up, right? So here is the experiment, because the theory has to be checked by experiment. So this is the biological age that I have just trained with the artificial neural network. These are the future treated and control groups. These animals were treated by experimental drug here, only 2 times, 2 shots. No drug in the circulation at no time after. And you can see that the treated group never catches up with the control. So if you do the machine learning correctly, you find the variable that is actually the aging variable, and this aging variable is defined in such a way, if you do everything right, that anti-aging interventions, short treatments produce lasting effect. So this is the rejuvenation. So once again, remember, in order to be rejuvenated, you have to be unstable. So this is, as usual in the world, the trade-off. So short-living species can be easily rejuvenated. Just one more example, like very old animals, time 0 here is 100 weeks old. These mice are geriatric mice. So you have 3 groups here. You have one group that is treated, the one group is the control, so they are dying from natural causes because they are too old. You have animals treated with rapamycin, so they are living longer, so they have mortality delay. And here we have one more group that is hit only once with 1 shot of an anti-aging intervention. So the rapamycin is given all the time, and here you have just 1 shot. You see the mortality delay here, and then 2 months after the treatment, you measure the hallmarks of aging in these animals. So these markers are correlated to the number of senescent cells, bad cells that are doing all sorts of damage. And you can see that the number of these, the level of those markers is reduced in the treated animals 2 months after the treatment, so the delay. So now you can go to humans. And as I told you, we have much more data from humans than from animals. And here is the biggest surprise. So if you do the same stuff, do this autoregressive, I mean, this is just a hieroglyph. This is autoregressive model, it doesn't matter. So it's a big autoregressive model. So you train the biomarker in humans. Remember I told you that in humans, your chances to die go up exponentially and double every 8 years. So instead, in humans, this biomarker oscillates. I mean, I cannot plot with Microsoft Office random fluctuations. So it randomly fluctuates, and then at some point late in life, it destabilizes and starts increasing exponentially. So humans turned out to be stable almost all our life. What actually happens is, I will tell you what happens later. So it turns out that humans are bored like naked mole rats. I mean, that's why we're a long-lived species. and only at some point, very late in life, we destabilize and become mice. Okay? So I'll show you in a moment how this works. So, as I told you, from the model, when we can actually see, since we have all the parameters of that model, we can measure the time that is required for a system to get back to the norm. Remember, if you're stable, you have the norm, you have the equilibrium level. So from the longitudinal data, you can measure how quickly animals get back to the norm. And we can see that the rate, the inverse time, the rate, is actually going down. So the rate is going down, which means that the time is going up. Remember, well, I can remember because I'm not that young anymore, but when you are 20, you can party on Friday and go and pretend to be working on Monday, right? And it works, actually. So elder people, not like they don't want to party, they just know that it takes a bit longer to recover from that. So the recovery time is a wonderful biomarker of aging. This biomarker doesn't exist in mice because mice cannot recover, as I have just shown to you, right? So humans can recover. So the recovery rate is going down, even in the healthiest individuals. And by the way, it doesn't matter what kind of data you are using. So if you measure this recovery time from blood markers or from your wearable devices, we did both, you can see that the recovery rate looks the same. You can measure it from any longitudinal data. So the recovery rate is the aging phenotype in humans. Basically, it means that if you can recover, it doesn't matter how aged you are, almost. But if you cannot recover, then you start getting all kinds of diseases and everything else. So other than the recovery rate, what also happens is that if you cannot recover quickly, then the same stresses get you further out of the equilibrium state, right? Because if the recovery force is bad, you are getting out of the equilibrium and you spend less and less time in your optimum state. I mean, that's what aging means. So if you plot the inverse variance, 1 over variance, in different signals, like in pheno-age, one of the popular biological age, in physical activity from wearable devices, from blood markers, it really doesn't matter. You can see that in all cases, the variance, 1 over variance becomes 0, meaning that the variance become infinite at about the same age, at about 120, whatever, 150 years. That means that even if you don't have diseases, aging is still there, which means that it's getting harder to recover, which means that you have more and more fluctuations of your organism state. You are not feeling good because you are never optimal, right? Because the fluctuations are larger and larger, which means that if we cannot control the mechanism that is reducing our resilience, our ability to recover, we will not live past 120 years old, even if we cure all diseases. So to make sense out of this, and then I will show you how to go to genes and how to go to the targets, I mean, visualizations are always good. So to make sense out of this, you have to think that there are 2 things that are associated with aging in humans. You can think about yourself, about your organism state as a kind of, ball rolling in some kind of, potential landscape. So this thing is getting kicked all the time. I'm eating too much, too less. I have a good mood, not too much good mood, everything else. So I'm fluctuating all the time. And my organism state somehow fluctuates here. And what happens is that I'm protected by some kind of a potential barrier here, so I don't want to go here, so that's why I'm alive. So I'm fluctuating here. But what happens, so basically this direction measures the stress. The more stressed I am, the more I shift it along this dimension. And then there is something else that is aging, actually, that is remodeling this potential in such a way that it becomes shallower, right? Remember I was showing you that the recovery force is going smaller. And this barrier, since this thing is becoming shallower, this barrier is disappearing. And at the age equals to about 120 years old, this barrier disappears whatsoever, and everyone is kicked out to the hospital. So I don't know, if it works, right? So what I told you can be visualized in this way. This is my happy life. Party, no party, party, bad party, death. Okay? So just enjoy this. I mean, it's good for a while. I mean, to make sure that this is not just a hand waving with the wearable devices, we actually measured the amount of people who have long autocorrelation time, for people who have very long recovery time. And it turns out that the fraction of the people who have the recovery time longer than a few months in the population is going up exponentially and doubles every 8 years. So actually the amount of people who are spending the rest of their lives here is actually increasing exponentially and doubles every 8 years. So that means that in order to become dead, you have to first become unstable. And the data, and it's really very new. It's only since a few years we have enough coverage with wearable devices, blood markers everywhere to check all those things. So it turns out that humans, we're talking about humans, we're all young here, so it's not relevant to this population, but on average we have a huge population where some people are here and some people are here. So we are actually two kinds of people. And you can only see this from the data. You cannot study it in mice because all mice are here, okay? So that's the power of the data, the power of things that you can do computation. So with this, I will give you two predictions, which you would be enjoying or not to follow, is that if you are a mouse, we have a solution for you. We can rejuvenate you, no problem. There will be infinite number of papers suggesting that there would be infinite number of drugs acting against infinite number of hallmarks of aging. All of them would reduce other hallmarks of aging and would produce lasting effects after treatments. Unfortunately, because humans are stable, stability means that both bad or good interventions cannot do a thing to you, right? So since you are stable, the very same drugs will produce small effects that are transient. If you are stable, it means when you stop getting the drug, your health state recovers to the original state. And I think, unfortunately, that's what we are seeing now in clinical trials in humans. Actually, all those drugs are working well in humans that are almost dead. In younger people, think about diets. We all know, I think, have experience with diets. You go on a diet, your weight is going down, your heart rate is going down, you stop the diet, you are back. Okay? Everyone knows that. The same would happen with the drugs, unfortunately, with most of the anti-aging drugs. So, well, as you know, this guy told you in the movie that there are two aging phenotypes. One of them is frailty, I mean that late life thing that happens when you get destabilized. And then there is aging, that thing that doesn't let me party anymore, right? So these are two things. Unfortunately, most of the drugs are studied in mice, which means that we study these drugs and we select specifically drugs that work in mice. They are not working here, or not working here very good, which means lots of the people who are studying aging without human data are here, most of the industry are here, and very few people are here. So we are looking from the data on those things that are controlling. We are calling that true aging. Okay, you cannot publish that in paper, so that's called thermodynamic aging. I will tell you why. But it's called TBA. It's like true biological age, okay? So we are acting, so I will show you now in a moment how from the actual human data to separate this and this, and how to find molecular factors that are controlling this and that. So now you would tell me, where are my aging clocks? There were lots of talks here about aging clocks, and I'm telling you something else, because the aging clocks are statements about the levels. So you have a level of certain marker that is up, and then my biological clock is up. I'm telling you something else. I'm talking to you about markers that are only measured along the trajectories, like the recovery rate and everything else. So let's get back to something that people understand, like DNA methylation and things like that. So that was computational biology. So I can go to the whole blood, and remember, at the beginning, I was selecting the clusters of features. I told you that when you are going to disintegrate, lots of features become correlated. So that's why you don't need to study specific features. You should try to find big clusters of correlated features and analyze how they behave. Try it at home. This is safe. You can take any human data set, and you would find that if you try to cluster them with principal component analysis, with variational autoencoder, or whatever you want, you would find at least two types of features in humans. So one of them would be linear in time, linear, and its variance would go up linearly. And then the other feature would be kind of parabolic in time. You would see the difference. And its variance would increase hyperbolically, and inverse variance would hit zero, as in blood, like in physical markers, at 120 years old. So which means that in DNA methylation, in proteomics, in transcriptomics, you would always find one feature that is associated with stress. This is this feature. I know that it's this feature because it's singular at 120 years old. And you will have the other feature that is linear in age that tracks the remodeling of the regulatory potential. So as I try to convince you, this is kind of true age in humans, and this is like stress age, right? So this correlates to Grimm age, for those who know, in DNA methylation, and this correlates to Howard clocks age. I can do the same in electronic medical records. I can do the same analysis, and I will find that in the electronic medical records, this is just a bit string. You have a disease or not have a disease, right? Zeros and ones. You would find that there is a feature that is kind of linear with age. And this feature correlates to the total number of the diseases. So this is just the disease burden, the total number of diseases so far. And the other feature would be correlated to the number of cardiometabolic diseases. So once again, one is kind of linear feature, the total number of diseases. I think you start guessing, right? So what is this true aging? Well, the true aging is just the total amount of damage that you have accumulated so far, right? And then all the other body components have to respect and reflect this damage, so they try to accommodate for that, and that's why you have other age independencies. So, what is this? I don't know, maybe I'll risk talk about this a little bit more. So, human bodies are sophisticated machines. So we have lots of components that can be switched on and on, like DNA methylation sites, different states of immune system and everything. And the total number, I would call them kind of functional units, right? So every unit could be on and off. And then by fluctuations, we're moving from on and off states all the time. And we have lots of such units. And some of them are soft units. Some of them are units that can change often. Like when I eat, when I smile, certain units switch back and forth. And there are lots of units that don't like to be changed, right? Those units are responsible for cell differentiation, things like that. I mean, those kind of very hard units. What happens as we age is that all of those units are changing their states. These states are changing very often. These are our stress units. These states don't want to be changed, but unfortunately we have fluctuations and some of them produce strong effects. So all the time, some of those states are pathological, well, most of those states are pathological. So if you have very many states that turned into the pathological state, then you have the effects of those transitions that are proportional to the number of those transitions. Since those transitions are independent, their effect is proportional to time, linear in age, and they produce a linear shift on everything else, and you measure it in blood markers. So that's how things work, like in disease markers in everything. So that's why in every signal, whatever you do, transcriptomics, methylation, in everything, you have a linear feature that is simply tracking the total number of damage that you accumulate over time, and the effects of those transitions produce the effect on measurable subsystems like immune system, everything else, that is linear in age. You see the effect. So basically, I'll jump here, and once again, this is just machine learning now, so everyone understands machine learning. So in order to understand how these things work, you have to go to large data sets, build, as I'm trying to convince you, autoregressive models. Here is where the computational biology is. You have the electronic medical records, for example, now. These are your diseases. Green dots are you have a disease, not have a disease, and so on. You have your electronic medical records in a few years' time in the future. And you connect the past and the present by an artificial neural network that is doing two things. First, as I told you, it compresses you all the diseases in a few clusters of diseases. It clusters diseases, right? Cardiometabolic health and all other. And then it estimates the biological age, which is the combined effect of all other diseases that are not clustered, right? So If I have a scratch on my hand, most probably it will reduce my lifespan because this is a pathological condition. But the more I have of those, the more of the biological age I have. So we have first the encoder part. So this is the network that is compressing the diseases into a few clusters of diseases and the biological age. Then we have an autoregression part that takes the state of health in the compressed representation and tries to predict the probabilities of activation of these units in the future, tries to predict if you are remaining healthy or not in the future, and then compares the predicted state with the actual state in the future. It's like the language model. In the language model, you try to predict the future symbol. In our case, we're trying to predict the future disease in every individual. And you can run this kind of models. Well, fortunately, we have Google and Facebook. They wrote TensorFlow and everything else. They educated infinite number of people in training these networks. As I told you, we're living in the best of the times, which means that all of that can be now implemented easily by a small company even, which means that you can run and train these models. What Kirill is sitting here, he is running and training these models. So you can run them. And if you ask Kirill, he will produce this kind of graphs for you. So first, the model infers the biological age. The biological age correlates to the total number of diseases, as it should be. Well, unfortunately, no, there is no magic. There is no magic, meaning that the more chronic diseases you have, the older you are. So the combined effects of those diseases actually reduce your resilience and produces the disease states. And then you can ask how many of those disease clusters of diseases are activated in the model. We can see that some clusters of diseases are activated very much, some not. Obviously, the one that is activated very much is exactly the one that has the lowest barrier. I mean, the weakest link. And this is obviously the cardiometabolic health. And if you look at the rate at which it is activated, this is exactly the mortality rate doubling time. So this model identifies the weakest link on the organism level. So this is all, of course, numbers. And with numbers, you can do magic. You can now combine the numbers, which we are getting here, the probability to get activated into a cluster, the biological age, everything, which we can now combine with genetics. I told you that we are living in the best of the times because now we have also the genetics for millions of people, not only the electronic medical records. Which means that once you know what to correlate with the genes, you can correlate with your favorite source of genetic information, and you can find genetic factors that are best correlated to your what is called phenotype. So for example, on the previous slide, I had the biological age. I can correlate now the genetic factors with the biological age. And I can see that, for example, in people that have this kind of genetic variant, the biological age is smaller. Wow. So probably we have a gene that is controlling aging, right? So according to Kirill, okay. So of course you would ask me, why would you need such sophisticated machines and why would you be doing this kind of machine learning? Because, well, we all know that what is longevity. We know that people have lifespans. Why not do genetics on lifespan? And the problem with genetics is that genetics is a relatively new technology. So I will tell you, unfortunately, you wouldn't believe me, but unfortunately, most of the genotyped people are alive, which means that we don't know their lifespan, which means that we cannot do genetic studies on their lifespan, right? Well, either we would have to wait, but it doesn't work because by the time they would be dying, there would be better genetics. And you would still not be able to do good genetic studies on those people, right? So fortunately or unfortunately, now you have to decide their parents are dead, right? And it's believed that at least half of the genes are coming from known parents, which means that you can do genetic studies on parental longevity, right? Because there is a correlation between your genes and lifespan of your parents. Which means that without these technologies, people were doing genetics of parental longevity in order to find genes that are controlling human aging. But the problem with that is that longevity is the effect of aging and diseases. So our parents had less access to medical technologies, and they were dying for somewhat different causes than us, which means that from parental longevity, we cannot know which factors are actually controlling aging. So if you do it the right way, you can scan all the genetic variants. This is the horizontal axis. Different colors are different chromosomes here. Every dot here is kind of genetic variant. And then sometimes you would find genetic variants like these bars that are associated with parental longevity. These genetic variant, these are known. This is the study that is published. This is a meta-analysis of 1 million genomes, so a very powerful study. So you can see this gene, this gene, this gene, this gene, lots of genes here are associated with longevity. So are they associated with aging or not? Who cares? So if you ask Kirill, he can do the same genetic analysis for the biological age, and cardiovascular mortality. And you can see that this gene from parental longevity study is actually a cardiometabolic gene, right? This gene is also nothing to do with aging. It's a cardiometabolic gene. This gene is cardiometabolic gene. Oh, this gene is actually from aging, right? This is from cardiometabolic and everything else. This, by the way, this gene is the target of a new drug against heart diseases. These are PCSK9 inhibitors that are developed by Amgen and Sanofi. So interestingly enough, if you do parental longevity, you have lots of genes that are associated with lifespan, but you don't know if they are associated with diseases or aging. If you do the machine learning, as I tried to convince you, you can actually delineate, you can separate the effects of diseases and the effects of aging. And now you end up living in an even better world, because now pharma needs to know which genes are associated with diseases, and for the God's sake, not with aging, because pharma doesn't want to do aging. And for the first time, instead of these, with modern longitudinal data, and remember that was longitudinal data, without trajectories you would not be able to do that. So for the first time ever, instead of this, you have this, and now you go to pharma and tell them, look, this is what you guys want, and this is what I want. So you can, for the first time, focus here on aging, and still be helpful to them to make sure that you address effects not associated with aging. So that brings me maybe to conclusion is that finally we can study aging in real world. We have trained engineers, we have lots of software, we have abilities to separate aging and diseases. We can understand the dynamic properties of aging markers and diseases. We can work with respectful companies on diseases to make sure that what we are targeting is not aging for them, because they don't like aging, and still work on aging ourselves. What this perspective is giving to you, in combination with all the modern omics, genomics, and everything else, it gives you molecular factors that are affecting the organism level phenotypes, like chronic diseases and aging. But what most important is that you are observing humans along their life history. And since you're observing them along their life history, you know what is causing what. Those models are causative. And you also know what is responding at which time. So you know that if you hit this, the response is tomorrow. If you hit this, the response is in 1 month. So you go to pharma companies and sell them everything that responds tomorrow, because they want to do diseases. And you still have full control on variables that are related to aging. So now I will give you just last punch, let's say the bad news. And the bad news is this. So what's the difference between aging and diseases? So, okay, I gave you some models, autoregressive models, variational autoencoders. They give you 2 numbers. Okay, one of them is aging, the other are diseases. It might be convincing. The problem is here. So remember I told you that there are lots of things that can get wrong in your body. Lots of them, much more than diseases in electronic medical records. That could be mutations, that could be some conformation transitions in your molecules, all kinds of stuff. The specific patterns of those things are different, not only in different people, but also in different cells. So each of the cells get damaged independently, which means that only the total number of those things is the same in different cells in one individual. The specific patterns are different. So all your cells are damaged in different ways, which means that it's very hard to think that you can come to this system with one drug that would fix 10 things in this cell without touching everything else, 9 things in this cell, not 10, because 9, this is another cell, and everything else. These are rare uncorrelated events, which means that this is essentially stochastic damage that is different in different cells. It's very hard to get one drug that would do all the things at the same time, which means that the true aging I mean, that's why we call it thermodynamic aging, is not really reversible. And, I mean, that's a bad thing. Remember, if you're a mouse, you can reverse aging. But that's easy because you're already exponentially disintegrating. When you're exponentially disintegrating, I can rescue you a little. Here in humans, we are dealing with a totally different beast. It's not a process that we can control. It's the result of infinite number of processes that together produce a linear feature. That means that with aging, unfortunately in humans, instead of thinking about reversing aging, technologically, we would be better off if we first think how to slow it down. Because if you slow it down, you can look at mechanisms that are controlling all of those transitions at the same time, like making a better living conditions for all the cells in your body. In such a way, you should be able to reduce the rates of all those transitions and to slow down aging. That's essentially the way to become naked mole rats. We believe that naked mole rats, I'm coming back to the beginning of this presentation, with all of that machine learning, lots of data and everything else, we discovered that humans are already almost naked mole rats. We are just not, our aging is not entirely arrested like in naked mole rats, but with all this machine learning and all the genetic data, we see the factors that are universally controlling the transitions other than diseases. We have separately the diseases and the aging, and still, as you remember, we still see genetic factors that are controlling what is remaining from aging when you subtract all the diseases. That gives us hope that we can see the factors eventually that would help us to arrest aging, at least to the level that it is arrested in naked mole rats. This is the only way to produce few-fold change in the lifespan in humans. Because as I just demonstrated to you, if you cure diseases, you may be living 120 years old, but your health state would be miserable still. I mean, the mere fact that you don't have diseases doesn't preclude you from the fact that you cannot party. So if you're not addressing resilience, you are not living longer. And even if you're living longer, you are miserable, which means that future effort should go into technologies that would be controlling this uncorrelated damage rather than stress. We will do something about stress in different ways. And as I try to convince you, you cannot learn about this from mice because they are dead within 2 years. They cannot teach you about aging. We are much more successful agers than mice. Mice have to learn from us, which means that we don't have, I mean, we're too different from naked mole rats, right? I mean, there are some differences, which means that we cannot unlearn aging from anyone else rather than humans. That's the punch. Unfortunately, with the advent of all these technologies and with more, how would they say, motivated or desperate, depending how would you count on these people who are working on this, we have a chance maybe to learn the factors that are controlling non-mouse-like types of signatures of human aging. And that was just a demonstration how you can do it. I think hopefully you can do it better. And well, here is the thing. We have some gene variants to test, and as usual, we need more data to find more variants, to test more variants and everything else. So I thank you for your attention. I hope it was not uninteresting.
All right, welcome. Today seems to be kind of the day of pessimism, so I'll try to give a solution to all these problems that we might not solve in time. A couple of these topics that I was asked to speak about, I have very limited to no idea what they're really about. So I'm going to focus on the stuff that I actually do know and quickly introduce the ones that I'm slightly less familiar with. To briefly introduce myself, I'm Emil, doctor by training. Back in the day, never really wanted to be a doctor. Did it to go into cancer research, which I then did after. Slight detour over running a few tech companies, and then after selling them, now finally back in the longevity space, with a slightly different angle than maybe the others. Running 3 organizations. One is a company, a biotech company that does human cryopreservation. One is a research institute in Switzerland that does fundamental research for the space. And then, of course, very fitting to this environment here, one is a DAO that funds fundamental research. So cryopreservation will be what I will be mostly speaking about, but then some other topics as well. I think if you talk about these topics where it's slightly less out of the mainstream maybe, and slightly more in the not fringes, but slightly on the edges of what is maybe total mainstream, I think it's always very important to remember that stuff that today is totally mainstream, like heart transplantation, hasn't been around for that long, right? And if you would have asked doctors in the 1950s, taking one heart from a human and putting it into another human, a lot of people would have said, hey, this is pretty weird. And of course, now this is totally standard. No one would want to miss it. Everybody's super happy that there's heart transplantation. And the same goes for planes and, you name it. So I think cryopreservation might be one of those, which today, for some people, might not be something where they might fully have to wrap their head around. But then, again, in the future, this might be absolutely standard and no one would want to miss it. And everybody would be like, hey, how could those in the 2023, in the 2020s, how could they not have had that, right? So, in addition, I think if you talk about longevity, I think it starts to be important to kind of look at the Venn diagram of what the space is actually talking about when they talk about longevity. A lot of people, of course, are interested in health span, right? Everybody is super down for living healthy, right? I don't think there's anything that's wrong with it. And then everybody who's into everybody who's into lifespan, like actually living longer, is of course also into living healthily while long. And then potentially when I say long, I don't necessarily mean like 5 years long or 10 years long, also 20 years. In my mind, this should be an individual choice how much someone chooses to live. If it's 80, that's great. But if someone wants to still live once they're 1,000, I don't think there's any superseding moral argument why they should not be allowed to want that. And if it's medically possible, do that. Of course, currently that's far from possible. So this is where, cryopreservation might come in. So my primary interest is always in lifespan. Not because I don't care about health span, just because, if you look at the squaring of the mobility curves, and I'll show you a couple of those in a second, then that is already ongoing, right? The mobility has been compressed. People get healthy, more and more healthy while they're older. But on lifespan, we've not made any progress in humans yet. Right, so I was always happy to say, hey, listen, I'll work on the thing where maybe my personal impact might be higher than running another lifespan company. And in fact, we ran a large study, and this is kind of similar data than all the other studies that have been run. If you ask people how long would they like to live, a good amount say pretty long. Right, in our data, it's 26% that says I would like to live indefinitely, maybe even forever. I'm not sure what the poll results from the polling thing here is, but I'm pretty sure it might even be higher in this group, but at least in this ballpark. Because this, of course, is the problem. Like, people have been getting, mobility is getting better and better, but everybody still dies. Somewhere between 95, and this has been true in the 1800s. This has been true, whatever, in the 1950s with all the advances in medicine, and unfortunately, it is still true. Of course, in mice, you kind of start to see stuff where, the intervention group actually does live longer, right? These red curves, they do live longer with some combinations. But then again, a lot of stuff. So all my research that I did in the past for cancer research always worked great in mice. I'm not aware that the people who followed after me did anything ever in humans, and a lot of stuff, of course, dies from the from the mice to humans translation. And you, of course, all know these curves, right? Or you might, the longevity escape velocity curves, right? Where currently we're kind of on this trajectory where everybody dies, and then with escape velocity at some point we might actually either get younger or at least stay young. The problem really is, this was published by Aubrey in 2004. Unfortunately, we're still there, right? We haven't moved this needle in humans at all. And we saw the slide in the talk in the morning today. It's just fucking complex. It's not like all these people are stupid. It's just fucking horrible. And I was not for I was a couple of years in the lab, and of course, unfortunately, if you've been in the lab for a few years, you really start understanding how horribly complex that shit is, right? And then again, it's not only that it's complex, also that it's very difficult to test, because you can't just do everything or anything in humans, right? So for better or for worse, and I'm of course one of these people who very much hope I'm wrong. I basically decided to go for cryopreservation because I'm more okay with being wrong this way around. If I'm wrong, then I'm just taking whatever pill there is in 20 or 30 years, right? Or treatment, do whatever it is, right? But I really, really, really don't want to be 85 businesses and die, and there's nothing, and the cryopreservation is still as bad as it is today, because there's still a lot of work to be done, right? So basically the logic why I've decided what this talk is kind of today about, and of course slightly advocating for cryopreservation, what if we're wrong? What if all this optimism that you read, like, in any paper or any like newsletter you read constantly, now aging is probably being cured, this new company is gonna do great. The problem is, if you talk to a lot of CEOs of these companies, internally they're not that optimistic. And even if we do it in 20 years, right, every day a lot of people die, right? And over 20 years, those 180,000 a day become a lot of people, right? Where basically, even if it's fine for all of us, there's still a tremendous amount of people dying until the time when maybe medical intervention are sophisticated enough to actually do something about it. So again, what if we're wrong? And we should start working on backups. All right, now a few topics that I'm not that familiar with, right? One backup might be something like hibernation. That's one of the topics. Everything that has a star on the top right is something that if there's someone in the room who wants to answer questions later about these topics, feel free. This is not my strong suit necessarily. So the idea of hibernation is that a lot of reasons why we're aging is because, well, we have metabolism, and a lot of stuff, the faster the metabolism is, by and large, the faster your heartbeat. There's this correlation between small animals, fast heartbeats, fast metabolism, die relatively young. Large animals, slower heartbeat, slower metabolism, maybe die pretty late. And then you can, if you hibernate, you reduce your metabolic activity. In squirrels, like super low, 99% reduction in metabolic activity. So you go really, really low in metabolic activity. And of course, the connection to cryopreservation here is fundamentally it's the same logic. Cryopreservation has the same logic, even though, of course, it goes much, much slower. I'll show you in a second, of course. So there is actually a good amount of like low-hanging fruits in quotation marks, what you can do with similar things, not really hibernation, but going into that direction, like hypothermia, in medicine today. There's like studies where you do in Baltimore where they treat gunshot wounds during operation to basically gain time for the surgeons to do the operation, reduce the core body temperature to not super low, of course, if I'm not mistaken, like 28 degrees or something like that. I'm not actually sure what the temperature is, but a couple of degrees to gain time for the operation, so that the brain and the rest of the body requires less oxygen during operation. And DARPA is funding mechanisms to bring soldiers that are being shot on the battlefield back to a field hospital to kind of bridge this time. And again, when you say bridge this time, right, fundamentally that's what cryopreservation does as well. Bridging the time from, well, dying until in the future medical technology has advanced enough, right? It's just a stopgap measure. It's not a solution in itself. All the other longevity stuff still needs to work at some point, but we might gain time to actually make it happen. Probably not going to be like this anytime soon, right? And this was one of the pictures when you talked about one of these hibernation things. From the Alien movie, where they have these cryopods, right, where they're being cooled down for star travel, which we will probably already maybe start needing once we go a bit further in Mars. Mars is probably still possible. Actually, there's a good amount of research. The European Space Agency is working on that, how to reduce body temperature to save oxygen, water, and food during transit, during flight, right? Maybe already until you get to Mars, but definitely if you go to the next star, you absolutely need it. Probably at that point, you actually do need cryopreservation. If you go a bit further in Mars, you might not need it yet. You just want to reduce temperature a bit. All right, but now let's go to the extreme. It's not this. This is always the pictures that I need to deal with if I'm talking to journalists. And then I ask them if they please can change the picture, and then they say no. Because of course, this is kind of what, like, freezing humans, freezing brains, that's of course how colloquially this topic is no, right? It kind of it's a relatively simple picture. Technically speaking, of course, we do everything to not freeze anything. It's cooling down to very, very, very low temperatures, somewhere between -140 and -196, so extremely low, to arrest all metabolic activity, basically have maybe atoms wiggle around a bit, but nothing is going on anymore. And then to bridge that time, and of course you want to do it without damage, you want to do it without ice crystal formation. Ice crystal formation would be one of the type of damages, right? If anybody of you has ever put like a strawberry in a freezer, and then took it out, let it thaw, then the strawberry there's no nice crunch anymore, right? It's now soup. Of course you want to very much avoid that with any brain, any human. And so you use specific type of cryoprotective agents, basically solutions that replaces all the water in the body against that solution, and that solution doesn't freeze. So now you can cool down to very, very low temperatures and keep the body at that temperature, potentially for decades, likely for decades, if not 100 years. I have no idea how long it might take. But to be fair, it doesn't really matter. Once you're at cryogenic temperatures, if it's 20 years, 30 years, 40 years, 50 years, 100, it doesn't matter. If it's 1,000, you need to make sure that the organization is still around, but medically speaking, even that would be fine. So why would anybody do that? Well, I think morally, as I said before, lifespan should be an individual choice. Unfortunately, it's going to be a good amount of time until that is medically possible. With cryopreservation, if someone dies tomorrow, there are some downsides, of course, but you can do it. You can choose this right now. It's always a last option. Right? I would never advocate for anybody choosing cryopreservation any day before they actually die and being pronounced. Basically, where you have the option, do you want to be cremated, or do you want to be buried, or do you want to be cryopreserved? I'm not religious enough to think cremation or burial is going to get me anywhere. Right? This is it. Cryopreservation at the very minimum, it's better than cremation or burial. Yeah. Right? It has a higher chance. The only downside, it costs money, and depending on your personal value system, you might feel like this money might be spent better somewhere else. But then again, as long as we're buying expensive cars and, do other expensive things, probably for most people, it would make sense to spend some money on cryopreservation, right? Done after legal death, right? Big advantage, all the FDA problems that other companies in the longevity space have, we don't have any of that. Right, because people have legally died, so we're outside of all, for better or for worse. There's some downsides to that as well, but we're outside of all regulation, and you want to do it very, very quick. As I said, it's a bridge to technology in the future to kind of maintain a chance that in the future, the underlying condition that killed you in the first place is curable, which I think at some point will be possible. Just like we cured infectious disease in the past. At some point, we're going to figure out cancer and heart disease and Alzheimer's and all the other ones. Just a matter of time, right? That needs to be, of course, happen. Then, if you're being cryopreserved, let's say at 85, of course, it doesn't make sense to bring you back if the average life expectancy in that day is 86, right? You need to have a good amount of remaining of life years left. But then again, I also think that will be figured out at some point. Again, just a matter of time. And last but not least, of course, you need to be able to reverse cryopreservation, which currently is not yet possible. It's very important to always state, this is always done with clear informed consent, meaning people who sign up for cryopreservation need to clearly understand, we don't know when revival will be possible. Technically, we don't know if revival is possible. So a lot of, it needs to be very clear that people know that they're signing up for a very experimental procedure that is a last-ditch effort, but of course there's precedent in the medical community for these last-ditch efforts, right? If you have terminal cancer, all these standard treatments have been tried, then patient and doctor can together, with informed consent, decide to do experimental treatments and so on. So cryopreservation, I would argue, is kind of just an extension, kind of like putting that concept to the extreme and saying, hey, if the alternative is burial cremation, then whatever other option there might be, everybody has the right to choose that. A lot of research still needed, and The cost, unfortunately, currently relatively expensive. So with us, with the organization I'm running, it's somewhere between 60 to 200,000, right? And then you can go a bit up. So relatively expensive, which is unfortunately prohibitively expensive for at least some part of the population, which is kind of like a pet peeve. I don't like this. There should clearly be a choice that such people make based on if they want to do it, not. It shouldn't be a factor of how wealthy you are, right? Luckily, at scale, cryopreservation can be extremely affordable, in the single digits, thousands of dollars or euros. But when I say at scale, it would be like, let's say 100,000, a few 100,000 cryopreservation cases per year, which of course we're currently very, very far away from. So how does this whole process actually work, right? So at the time of circulatory arrest, but not before the person has legally pronounced, right? We want to start as quick as possible after the heart stops beating and the body is not supplied sufficiently with oxygen anymore. Because at that point, the death process, right? It's not really a point, it's a process. Body stops, doesn't have oxygen anymore, and now cells start to degrade with a variety of apoptosis and necrosis and so on, right? This process you want to first slow down and then stop. So the first step of this process is what is called kind of standby and stabilization. So we dispatch, when we know that someone might die, we dispatch one of our medical teams to the patient. They wait in front of the hospital. And then the second the patient dies, we take over and now start to cool down the patient with a variety of methods and give cardiopulmonary support. So chest compressions, what you would do for CPR. But of course, in our case, the goal is not resuscitation, but just supporting the cardiopulmonary function to supply the brain with oxygen and make sure that the brain stops that degradation, that cellular dying process as quick as possible. Once you've done that, you now replace all the water in the body against the cryoprotective agent. Basically, we have heart-lung machines. We have mobile, I'll show you in a second. We have mobile heart-lung machines. We do median stenotomy, so open the chest, cannulate the heart, and then perfuse, just like you would with blood in an operation room. We perfuse this cryoprotective agent, circulate it around the body, also a coolant, so it also cools from the inside. But more importantly, it allows us to now go under freezing temperatures. Right now we can cool down further and reduce metabolic activity even more. So that's what we do. Second, at that time, we then transport the patient to, in most cases, to our Swiss Research Institute, where there's going to be CT scan for quality assurance and so on, and then very, very, very low cool down to cryogenic temperatures. You do this very, very low cool down because once you cross, in fact, it's not slow in the beginning. First, it's very fast. So you want to cool down to around, once you have the cryoprotective agent in, of course, you want to cool down to, let's say, negative 120 as quick as possible. So we have specific like cool down machines that we designed that are like human sized, where liquid nitrogen is like a liquid nitrogen vapor is like is circulated, so you can cool down something very, very quickly. And from negative 120, you cross what is called the glass transition temperature. Basically, the body goes through a state change. You know, it's not liquid anymore, kind of. It goes into a solid-like structure called the so-called the vitrified state, right? So it basically becomes like a glass-like amorphous structure. So think of it as kind of like from a property standpoint, like glass. It's a solid now. And now you want to cool very, very slowly at that point, because otherwise, if you would cool faster, different parts, most importantly, the brain, of course, different parts would cool at different rates. You know, the outside would cool faster, the inside would still be warm or warmer. And if that happens, you generate thermal stress. And this thermal stress can lead to what is called cracking or fracturing. So just like you might remember, like ice cubes, they have these fractures in them. Like basically, they're not fully transparent. They have like fractures, and this is similar-ish looking to that. Now, the slow cooling allows to always give time to have these thermal stress released. So we do at least 100 hours. So from negative 120 to negative 140 or negative 196, we cool around over 100 hours. The reason why I'm saying negative 140, this is a new technology where you have higher storage temperatures to not have that fracturing happen that much, right? Well, and now we wait, right? We might wait 50 years, we might wait 100 years, it might be longer than that, until you can then have the technology to repair all the damage that has been caused, and then potentially revive the patient at a time when in the future, all these problems are solved. So I kind of give you a visual impression. So this is one of our medical ambulances. We have a couple of them in Europe. We're now starting in the US as well, at least in some metropolitan areas. There's always doctors, perfusionists, a couple of people who are trained by us that actually, trained for cryopreservation. This is kind of how it looks inside. Basically, they're mobile operating rooms. They're not really ambulances anymore. They're kind of mobile operating rooms with perfusion systems. So this is one of those heart lung machines that is being used to put the cryoprotective agent into the patient, and then. As I said, they're being brought to Switzerland. I'll give you a bit of more of a visual feel, right? CT scanner, operation room, and so on. And then the patients are being stored in these kind of like 10 feet high, 3 meters something high, stainless steel, so-called cryogenic dewars, filled with liquid nitrogen, either liquid nitrogen or the vapor phase above liquid nitrogen for these different storage temperatures. This is all underground, so for maximum security. Basically, this is the building here under the building here in the back. This is where this the storage room that you just saw is, and then of course there's also lab space to doing biomedical research. Now let's say we're in the future, right? What part do we actually need? The body, probably less important. Right? The brain, of course, is the important part here. Of course, we don't fully know what exactly we need because, where consciousness and so on comes from is kind of still a bit of an open question. What we probably want to have is the connectome, so the connection between all the cells. So, of course, you have a lot of, neurons in other types of cells in the brain. All of the connections you want to keep. You want to keep the weights of those connections, right? Not every connection between two neurons is the same. There's different. They have different excitation, how they're being excited and so on. How strong the connection is, which is probably partly encoded in the synaptic clefts. Like what kind of neurotransmitters are there? What kind of transporters are there, and so on, and probably a good amount of other factors. So you want to keep all of that, and probably you want to keep a part of the internal setup of the cell, which is kind of the computation. What kind of computation happens in that cell? What kind of processes are running in that cell? Cryopreservation keeps these two things pretty well. For computation, it's not exactly clear what exactly is needed. Some of which would be well preserved, other stuff less so. So depending what turns out later to be important. So we try to keep or improve the process so that more and more of these things are preserved. So there's basically the state is as close as it is while people are alive, and then hopefully that is sufficient in the future to make it to revive people. The rest of the body probably. So one slide about synthetic organs. The rest of the body you can probably in the future either with organ 3D printing. With xeno organs like pig organs that are humanized, with other types of technology, probably replaceable, right? Because we're not talking about 10 years in the future, we're talking far in the future. And of course, there's a lot of economic pressure to make these things work just for normal, in quotation marks, medical progress. So probably it's possible to do that, right? There's still going to be a good amount of repair needed. If anybody's interested, we can discuss in detail. Otherwise, there's a very extensive book from Robert Freyes called Cryostasis Revival. It was released a while ago. Robert Freyes, one of the nanotechnology experts from the US, apparently started out as a paper. Someone wanted Robert to write a paper, and now it became a 700-page long paper kind of thing. Extremely detailed, a lot of math. So if someone really wants to dig deep, not an easy read, but very exciting, a lot of cool information, basically outlining how revival would be done in detail, how in excruciating detail, how all the damage that is currently happening during cryopreservation, how that would be repaired in the far future. Now, after that, right, now let's say revival is possible, right? You would do this repair, and then you would revive someone, and then they might live on in the future. Now, some of you might say, well, I don't want to live in the dystopian future, and for that, I think it's always good to remember all the high-level metrics of like poverty, people living under the poverty line, children dying, and so on and so on. These are just a couple. There's more of those. Over long horizons, everything has been getting better. Sometimes a bit difficult to see because, of course, these lines are zigzag lines, as you can see, right? Some are very, very zigzaggy, right? So it always feels like we're living in a horrible time, but over long periods of time, if you just look a couple of decades or 100 years back, we're living in the best possible time. There's not a single time in the past where I would rather, switch to. And then now, I'm probably not sure if these arguments will come today, but if people think of overpopulation or how are we feed all these people, or how is going to be the energy, how are we going to, have enough food, If you take vertical farming, if you put that size with solar cells, right, or fusion reactors probably is way less space than that. You can easily, with new technology, probably keep everybody alive in the future. And then again, currently, the amount of people who have cryopreservation contracts, so not cryopreserved, but cryopreservation contracts, is less than 10,000 people. Even if we're adding 1 million people in the future, right? 1 million people out of then 10 billion, where we kind of got to level off probably worldwide population-wise, it's nothing. It's negligible, right? Even if it would be 100 million, right? That would be 1%, right? Absolutely negligible in additional people. And of course, at that point, 100 years in the future, some people might already have migrated to Mars or to wherever, or I don't know where, but probably overpopulation, all these problems are not going to be a big issue. Now, this is kind of revival in the flesh, right? So basically, you keep your body, some parts might be replaced, but part of the community is, or kind of like a thought, is not necessarily that you would be revived as your body is right now, but rather be re-uploaded, right? So basically where your consciousness would be reinstantiated in silico. Basically, you would do all of these things. You would map the connectome, you would map the weights, you would map the computation, and then you would basically reinstantiate the same processes that are currently running in your brain in silico, in a computer, right? Or whatever the substrate on which that computation then runs, whatever it would be. The logic now, of course, would be, or the assumption would be that as long as your processes run, then this is you. There's some philosophical problems, questions that we can discuss over wine tonight. I'm not super hot on uploading for a variety of reasons, but other people are super down with it, and I'm not sure if I'm right or if they're right. Now, to leave a bit of time for questions, let me really briefly run through a couple of... should I start, or do you want to ask a question now? No, go ahead, please. Absolutely.
So, how many people already in Cryo preserved the real pulp?
Yeah. So we've done around somewhere between like 15 cryopreservations. So we're the youngest organization out there. Currently, we only cover Europe, so we only have members in Europe. Next year, probably we're going to be the biggest by people who have signed up. But since we have relatively young people who sign up, average is 37 years, statistically, they're going to take some time until they die, hopefully.
But they pay that they sign, so they pay that they die.
So they pay 25 euros about per month. So basically, there's a membership with us, right? And at the time of death, you pay either 60 or 200,000. So you don't need to pay early. We actually don't want you to pay early because we can't keep that money. It's your money, and so it doesn't make sense to pay early. So $25 plus at the time of death. And you can take out life insurance, right? A lot of people fund that with life insurance. We have a large life insurance partner where you give them another $25. And then if you die tomorrow, they pay the 200,000. Assuming you're relatively healthy and relatively young and so on. So a couple of small print, but in general, yeah. Then a couple of these people who have been cryopreserved, we do a lot of body donors. So accept body donors where they're not being stored long term. They're just like you would donate your body to a medical university. They donate them to us so that we can train our teams.
Okay.
All right, science. So what is already possible? So you can cryopreserve organs, right? This is one of the landmark papers from Greg Fay, where they cryopreserved a kidney, a rabbit kidney, and then put it back into the rabbit, and it worked, and so on. You can cryopreserve certain organisms, like C. elegans, right? Keeps its memory. So after being revived, it still has its memories. I'm the first to admit that C. elegans memory is probably slightly different from human memory, but in principle, it shows that the neural connections, all these weights, all of that stuff that we would want to keep around, that in principle, this is possible to keep around. And then another paper with the slight variety, or this was done with the slight variety of a cryopreservation, so-called aldehyde-stabilized cryopreservation, which is cryopreservation combined with chemical fixation. You can show that the brain structure is exactly how it's supposed to look. Neuroscientists can't differentiate these two. So you can show that, as far as we understand, the part that makes you, and me, that part is being kept around. Right? And then the logic is, as long as that is still around, for argument's sake, you're kind of still around, or at least you could be around in the future. But there's a tremendous amount of work still to be done, right? Like cooling is a problem, warming is a problem, you need to do it very fast, cryoprotective agents, you need to reduce the toxicity of them, and so on. So there's a long list, right? Ischemic damage, so basically the time when the body is not supplied with oxygen well enough. It's called ischemia. During that time, damage is happening. There's still some ice nucleation. So when ice crystals form, they don't directly form, they nucleate. So basically they have a starting point and then they grow around that starting point. So that's still happening to a degree. There's thermal stress, as I discussed, during this cool down procedure and so on. And there's a long list of other topics that would be out of scope to discuss right now. A good amount of that stuff, a couple of these things, like on the last slide, these things, we're working at these are not low-hanging fruits, but these are tangible things that you can almost work with engineering type of approaches. So with those, we're working either at the foundation or at the company. We're trying to make them better right now. Here, this is the long list of problems, where some are very moonshot, like high impact, but also very difficult to do. And probably it's not a comprehensive list. There's probably a good amount of others or more details. But for those, the third organization that we have founded, actually with a couple of people from the VitaDAO crowd, who's also here, like Lorenz and Vincent and so on, is like a DAO that will do half, kind of like VitaDAO, not low-hanging fruits, but relatively low-hanging fruits, stuff that is commercializable relatively soon, that might generate revenue, right? Either for cell preservation or tissue preservation or organ preservation or organ biobanking, right? About half. And the other half will go for hardcore, like long-term moonshot kind of things that if they work out, they would really be game changers. Like, for example, enabling organ biobanking, where then the organ shortage would be significantly reduced if you could keep organs long-term in all the biobanks, right? And of course would also help cryopreservation. So these are the two first research projects, just to give you a bit of an idea. One would be, this is kind of like a moonshotty kind of thing, ultrasound heating. Basically they build like large arrangements of thousands of ultrasound transducers that are inside of an MRI, where they're all like slightly offset, where you then in one pulse of these ultrasound emitters can warm up a large volume uniformly and outrun the ice crystal formation during warm-up. If anybody's interested, I'm more than happy to discuss further. And the lower-hanging fruit stuff that can be soon done is like a new combined medication therapy to improve the cryopreservation procedure. So, last but not least, the partial reprogramming is going to be great if it works. If it doesn't work, there was a time when we thought we just need to do telomere elongation. Hasn't worked out. There was a time when everybody thought, hey, calorie restriction is the shit. It probably hasn't worked out, right? So there were a couple of times I feel like this field goes from hype to hype to hype, and we always go through the Gartner hype cycle. And at some point we go through the valley of disillusionment, and we're like, hey, maybe it wasn't as easy as we thought. And of course, me coming from the cancer field, well, the cancer field has been saying for the last 40 years, it's just another 10 years. Just a couple more billion and just a few more years, and we have this figured out. And it's not anybody in the cancer field, right? In 2005, the director of the National Cancer Institute in the US said, the director's challenge is to cure cancer, not make it better, cure by 2015. Well, we're a couple years over and we're not even close to that, right? So I think it's much better to be wrong this way around, where if all the economic pressure is anyway on making, whatever Altos and whatever there is to make it work. But then again, there have multiple times been in the past when, like, all these very optimistic, ideas have not turned out to be true in biology. And probably that problem has been exaggerated by the fact that more and more tech people who are used to significantly faster innovation cycles have gone into the longevity space. Right? And they're used to kind of like the tech innovation, which has always been faster than the biological innovation. As I said, as I saw on the slide before, it's really hard. So I think working on backups is a really, really good idea. So if anybody wants to discuss more, we're probably going to have a longevity plus cryo dinner sometimes in the next days. So join us. I don't know the date yet, but we're going to figure it out. And for now, I'm more than happy to answer any and all questions. Please. Please.
I'm sort of like